Okay. So now we move on to, before we go on to central dogma again, let me just go through here. This here is the nucleus. These are the nuclear pores through which substances can come out or exit and enter in. And this whole structure is the cytoplasm. Okay, this is cytoplasm. This is the nucleus. Inside the nucleus we find a dense portion known as the nucleolus. This nucleolus actually is responsible for producing rRNA or ribosomes. Okay? It does not produce ribosomes straight away. It actually produces rRNA, this part, nucleolus. And it also produces tRNA. And tRNA are the shortest RNAs, whereas RNAs, they are a little longer. Okay? And then mRNA is produced outside the nucleolus, but inside the nucleus. Okay? So this is the mRNA, and mRNA moves out. But before mRNA is formed, tRNA and rRNA, they are already produced by the nucleolus, and they are already outside the nucleus and in the cytoplasm. And in the cytoplasm, where splicing occurs, after the splicing of rRNA, it forms the large subunit and the small subunit. And these two join to form a complex called the ribosome complex. The large structure here and the small structure here. We saw that the small structure is responsible for attaching itself into the mRNA, whereas the large subunit of the ribosome is inclined towards grabbing hold of the correct tRNAs in order to fix it to the codons on the mRNA. Okay? So that is what happens of the RNAs and the tRNAs. We find that mRNA is formed at a later stage than RNA and rRNA and tRNA. Now let's move on to the central dogma of molecular biology. At last, this is nothing but central dogma. It comprises just three terms: transcription, translation, and protein synthesis. Actually, four terms. Okay? Transcription, translation, and protein synthesis. I'm going to start with its site. Where does it take place? Transcription takes place inside nucleus, and translation and protein synthesis occurs in ribosome. This is inside the cytoplasm. Okay. Now transcription is different. Transcription is, this is DNA. Here's another DNA. DNA replication means all of this DNA is copied, whereas transcription means only a section of the DNA is copied. So this is transcription, okay? Just copying of a short portion of the DNA, whereas the copying of the entire DNA is known as replication. That's something else, which we shall deal with in a later video. So let's start with transcription. It involves three important steps. First, it's initiation, elongation, and termination. So I don't know whether we'll get through initiation today, but let's just try. Initiation is the process where RNA polymerase II binds to the promoter region of DNA. Okay, let me just make it simple. Here is the DNA that needs to be copied. Okay? What this sentence is saying is RNA polymerase. Now, RNA polymerase is an enzyme that actually helps form mRNA. RNA polymerase II forms messenger RNA. Okay? So when this particular enzyme, known as the RNA polymerase II, comes and fixes itself on the DNA that needs to be copied, it needs to know which is the portion that needs to be copied. So in order to do so, there has to be a code which the polymerase, RNA polymerase can read. So it's called the Tata box. Tata box because it has a DATA. Okay, so this can be written like this: DATA and AA. So those are the 6 codons. This is called the data box. This data box is called the promoter. So let's say the data box is here, okay, and the RNA polymerase 2. So this here is a diagram of the mRNA, and actually this is not the mRNA. I made a mistake here. This is actually the DNA. It does not have a cap, etc. This is the DNA. Okay? I made a mistake in this diagram. It does not have a tail. Okay, this is the DNA. It has, instead of data box, it does have data box, but it has exons and introns. So this is the structure of the DNA. It does not have a cap and a tail. Okay? So this is the DNA which needs to be copied, and so the RNA polymerase has to come and fix itself on a place from where this copying needs to be done. So this code, known as the data box, tells the polymerase to fix itself somewhere around here. So once it does so, let's look at this diagram. Here you have the DNA here, and then the data box where the RNA polymerase 2 fixes itself on the DNA. Then it begins to split up the DNA by removing the hydrogen bond between them. So this is what happens. Data box, and then the RNA polymerase has moved towards the 3' end. Let's just say towards the opposite end. Okay, so it's moving towards the right side, and it has already opened up this, the DNA. Okay, so now the DNA can be copied. So this is what initiation is all about. Initiation is the formation of a Y fork. This is known as the Y fork. Now, from here, once the Y fork is formed, only then the mRNA can be produced by copying. So how this is copied is we shall discuss in another video that talks about elongation. Okay, it needs an entire video on it. So let's just settle here. Just understand that initiation is the process where the RNA polymerase 2 fixes itself just behind the data box. What is the data box? It is the promoter. What is the promoter? It is the place where it is the place which tells the polymerase that the DNA codes from then on needs to be copied. Okay, but the copying site is what do you call it? Guided, so to speak, by the data box. And the work of the polymerase is not just to add nucleotide sequences. It's not just to add nucleotide sequences, but also to split open the already bound DNA into a Y fork from where the DNA can be copied. Right.